With the world still grappling with the original variant of COVID-19 that was first found in the Chinese city of Wuhan, a number of other mutations have emerged with concerns that there could be more to come. Experts say the longer the virus is allowed to spread, the greater the opportunity there is for it to also mutate. South Africa currently battling the deadly Delta variant, and it's also detected 3 other variants of concern. So how does a virus like SARS COV-2 actually mutate? For this discussion, let's bring in Dr. Thovowani Ramulongo, who is a microbiologist as well as a researcher at the CSIR. Doc, it's great to see you. Thanks very much indeed for making time to chat to us. Let's perhaps begin with the elementary knowledge and speak generally about how and why viruses mutate to begin with.
So viruses like coronavirus, where the genomic material is RNA, when they make copies of other, when it replicates and make copies, it makes a lot of mistakes. So when it generates mistakes, it does not go back and double check, which in turn it ends up generating copies of these viruses that are slightly different from the original virus.
There are hopes in our case that something like a vaccine might be able to help us, in some respects, curb the continued mutation of the virus. But how does that happen in a context where we know that vaccines don't necessarily stop the virus from spreading?
Definitely, the vaccine will help a lot to curb this pandemic. What happens? The virus is it's like a ball. So on the surface of the ball of the virus, there's spikes. So the immune response it's generated highly to the surface of the spike. So, but the problem with the spike, there are regions that changes quickly, but also there are regions that are highly conserved. These regions that are highly conserved, the virus needs it to enter the cells to set up the infection. So when the immune response reacts to the virus, usually it generates a high amount of antibodies towards the vulnerable regions, but also to the conserved regions. So in the case of vaccination, the immune response will have antibodies against the vulnerable regions and also the conserved region. So when the virus changes, and then an individual is exposed to another variant, there are still antibodies that recognize the region that's conserved that the virus needs to enter the cells. So those antibodies then help to fight the new variant at the early stage of infection. So even though the vaccine at this point, they don't protect 100% against different variants, but it also reduces the severity of the infection.
We have heard about the Alpha variant, the Beta variant, and the Delta variant in our context here in South Africa. We're told that this third wave is driven largely by Delta, and it's almost gotten to a point where we're not able to detect Beta and Alpha. Speak to us about how different variants of one virus interact, if at all.
The different variants, like the small changes that happens on the virus, it can give the variant the advantage to escape the immune response, or it can also give the virus the opportunity to spread even further, even faster. So that's what we are seeing with the Delta variant. So the Delta variant, it has the capability to spread faster.
Right.
So that's why we're now seeing that we have more of the Delta variant.
And when a virus is mutating, is it usually trying to balance the ability to spread faster with the ability to be more deadly? Because again, we'd found that Beta perhaps doesn't spread as fast as Delta, but Beta is a whole lot deadlier in some respects. In other words, actually, it's able to bypass immunity from the vaccines we have. However, Delta is able to spread faster, but we've seen even AstraZeneca, for instance, as a vaccine, is still able to push back against it. So Is that usually what we see typically? A virus usually decides whether it'll spread easier or whether it'll be more deadly, or can it be a combination of the two?
So actually, for the virus, it's actually a case of its survival. So the virus is not necessarily trying to be deadlier or be more transmissible. So what happens, this virus, when it replicates, it makes errors, and these errors, they are random. It's just that in a population of viruses, as it replicates, there will be a population that will be selected, and that such is a population that has more ability either to replicate or more ability to enter the cells or more ability to escape the immune response. So these things is the things that are happening by chance, depending on the environment where the virus is growing itself, is replicating itself.
That's right. And are we able to simulate how a virus is likely to mutate next? I mean, I understand you've just mentioned that usually the mutations are quite random, but in a lab with some of your colleagues, is there a way of trying to, dare I say, even guess what the next variant might even look like?
It's difficult to be able to predict what the next variant might look like, but it's also possible. It is also possible to be able to tell where the regions that accommodate mutations, like what I said, with the site that the region that the virus needs to enter the cells. So that region the virus needs it, so it doesn't change it. But there are also regions where on the capsid itself where it allows changes. So the more we study the genetic material of these viruses and also trace the mutations that are occurring over time, we can be able to predict the regions that are more susceptible to changes. And therefore, these are the regions usually that leads to change in variants.
Dr. Thovowani Ramulongo, thanks very much indeed for your time. Really appreciate your insights this morning. Dr. Ramulongo is a microbiologist and a researcher at the CSIR. Thanks very much indeed.